Hey guys, welcome to today's video. We are talking about mutations. But before we get into the learning goal, I wanted to recap what we did last week. And so here we have a GIF from the Amoeba sisters showing us the process of protein synthesis, whereby there's two steps that take place. The first one being transcription and the second being translation. So here in transcription, we have DNA. RNA polymerase comes in, makes a copy of messenger RNA. That messenger RNA joins with a ribosome. Transfer RNAs come in, drop off their amino acids, and a polypeptide chain then folds upon itself to create what we know as a protein, a functional expression of that original genetic code or gene region of DNA. So the learning goal for today, firstly, is to identify how mutations in genes and chromosomes can result from errors. That can be in DNA replication, or it can be in cell division, or even caused by mutagens. We will explain the process of non-disjunction, and when we see the cognitive verb explain, that's a pretty lengthy response that's required, so you're looking at about 5 to 8 sentences. We will look at karyotypes again, and lastly, to describe about 3 to 4 sentences worth of how inherited mutations can alter variations in genotype of offspring. So let's get into it then. This is from 10.1 of your text, and again, the key terms are down the side here for you guys to read more in your own time. So nature does an amazing job at replicating DNA, undergoing mitosis and meiosis and synthesizing proteins, but rarely makes mistakes or mutations do occur. Mutations are simply changes in DNA. If we have changes in DNA, this will cause changes in the mRNA, and that then in the amino acid sequence, and that will cause changes in the final protein. So anything with DNA can experience a mutation. That includes animals, fungi, protists, bacteria, and you and I. And as you can see in this image, here is a phenotypic expression of a genetic mutation that has occurred. We can have changes in DNA at a single point, and these are called genetic or point mutations. Or we can have changes in chunks of the chromosome, and these are called chromosome mutations, and we will look more at these on the next slide. So there are 3 types of genetic or point mutations that we are going to talk about for now. And the first one is called substitution. And if you can see here, there in the top of the image, we have the original sequence. Below that, we have a base substitution that has occurred. So instead of there here being a thymine, we have a cytosine represented by the C. This type of point mutation, and it's called that because it occurs at a single point in the genetic code, may be what we call silent, because if it changed the code but not the amino acid, then there's no real difference in the end in the final gene product that is produced. However, if that single point substitution does change the final amino acid, and that could have a larger effect on the protein in the end, then we don't call it silent, we call it missense. And I'll show you an example of this. Here's a codon chart, and we can see here that the way we read it is, all right, if there's a C, a U, and a, let's go over here, a U, well, it codes for leucine. But if we have a C, a U, and a C, again, it codes for leucine. If there was a change between CUU and CUC, it would be silent. There's no change in the final amino acid. If there was, then it could change the protein and therefore either be non-functional or even harmful to the organism. The second we see here is an addition, a base addition, or as the textbook calls it, an insertion. Insertion and underneath that, base deletion, these can be even more significant because they have the potential to be especially dangerous by causing what is called a frame shift. And since bases are read in threes, if we add or change a base, delete a base, this causes the whole reading frame of mRNA to change, thus possibly affecting the whole following amino acid sequence. You can check the description of this video for a cool animation just showing that graphically. There's the three types, and if we were to recap this using one of the Amoeba Sis's GIFs, it would look like this. So we have, I'll just move this down here. We have insertion and deletion up the top here, and as you can see, that would change the way in which this code is read. So instead of that being the 3, this would be the 3, causing a frame shift, therefore possibly affecting a huge amino acid sequence if this mutation occurred in a gene coding region. If this mutation occurred outside of a gene coding region, it wouldn't really matter. It would just be a change in a non-coding section of DNA. But then we have substitution below here. And again, the two types that this can result in, or two main types, that would be either silent or missense, depending upon if it changed, if that codon changed the amino acid or not. Okay, so 10.2, we can also have large scale DNA changes, and this can happen in chromosomes. It may occur during replication, for example, where instead of one gene or one point being affected, we can have a whole portion of a chromosome changing, therefore affecting hundreds or even thousands of genes. Let's look at a couple of examples of these. So firstly, duplication is where a portion of the chromosome is duplicated, and we can see that in what's represented by this B section of the chromosome. Deletion, as the name suggests, is where part of that chromosome is deleted. It can break off. We can have inversion, where that part of the chromosome is replicated, but it's flipped. So we can see A and F are staying the same, but this whole region in between here has been inverted. And lastly, this translocation, and that's where a fragment from one chromosome breaks off and attaches to another chromosome. There's many different types of mutations that can occur in chromosomes, but these are just a few, and hopefully that gives you an idea of what can take place. There's especially vulnerable times where mutations can happen though, such as during mitosis and during meiosis. And just to recap, here's an image of, we're looking at a cell and the genetic material that is lining up along the metaphase plate during the stage of metaphase. That then in the next phase, if you remember PMAT, is anaphase, and now that genetic material is being pulled to either poles of that cell by the spindle fibers to what we call the centrioles, these two ends. So that's just a bit of a recap to give us context to not that image, this image here. So during meiosis, though sometimes chromosomes don't separate completely like they should in this stage, what is called non-disjunction can occur. And this can result in an egg or a sperm cell that has too many or too few chromosomes. So the presence of an abnormal number of chromosomes in a cell is called aneuploidy. And here is an example of trisomy. This occurs when an organism that's meant to have 2 chromosomes has 3, and we can see that in this karyotype image on chromosome 21. So when there is an extra chromosome 21, this results in Down syndrome. It was first discovered in 1959, and it's not the only case of trisomy. If you check out the textbook, there's a few more examples of that in there too. So let's look at this diagram now. Non-disjunction, which results in aneuploidy, can occur either during meiosis 1 or meiosis 2 of sex cell division. So in meiosis 1, chromosomes, homologous chromosomes, fail to separate. So we can see there's these chromosomes that are lining up, but in non-disjunction, these homologous chromosomes have failed to separate, causing an increase in chromosome numbers in this cell and a decrease in this cell. But in meiosis 2, we're looking at this stage of division, and this is where chromatids, sister chromatids, fail to separate. So to recap, homologous chromosomes fail to separate in meiosis 1, sister chromatids fail to separate in meiosis 2. Either way, there is an imbalance in the final result of gametes. We can either have n+1 or n-take-1. And remember, gametes are what are called haploid. So this n, say if this was a sperm cell, would join with the female ovum, and that would become 2n. So there would be a set of chromosomes from mum and a set of chromosomes from dad. Let's look at another amoeba sisters GIF, just showing what we spoke about in this first section here. So we can have duplication, deletion, inversion, or translocation. Changes in chromosomes. I'll let that play through another time. Okay, let's keep going. Lastly, 10.3, causes and effects of mutations. So mutations are random, but we can have factors that make mutations more likely to happen, as a lot of mutations are caused by environmental conditions or exposure. Blue eyes, for example, are believed to be the result of a mutation that took place in people who migrated to Africa from Africa to Europe many generations ago, as it increased vision in poor lighting. And this photo is pretty cool because it also shows an example of what we call heterochromia. And in this case of the iris, the part of the eye, the colored part. And in this set of twins, this little girl has 2 different colored eyes. It's a variation in coloring of pigment between the 2 eyes of an individual. And maybe you know someone who has that as well. So genetic mutations can either be inherited from the sex cells of a parent, which are present throughout the entire life of an organism and are passed on to every cell and to the next generation of offspring. If not inherited, mutations can be somatic, and this means non-reproductive. So somatic means body. We have sex cells, X and Y, and then we have sex chromosomes, X and Y, and somatic chromosomes. That's the rest of them. We also have sex cells, gametes, and we have somatic cells. That's the rest of the cells in our body. So if it's not hereditary, it can be somatic, and this means that the mutation has been acquired over the individual's life, possibly due to either random chance of things happening during replication or due to environmental exposure to a mutagen. And a mutagen is a physical or chemical agent that causes change in the genetic material of an organism. It can be chemical, and an example of this is benzopyrene. It's the chemical that is found, one of the chemicals in the tobacco of cigarettes that affects and binds to the DNA, the genome of an individual. It can also be energy or ionizing radiation, and they are highly charged subatomic particles such as alpha, beta, and gamma, which passes through cells and tissues, cutting up the DNA in its path. It can rearrange entire sections of the chromosomes, altering relatively long stretches of DNA, and this is what causes cancer. Cancer is just uncontrolled replication of mutated sections of DNA. And Marvel based their Hulk comic series on this idea, in which Bruce Banner exposed himself to high levels of gamma radiation, therefore transforming his genetic genome and his phenotype into the Hulk. Here's an image, probably a more realistic one now, of maybe you recognize this. It is the structure, the dome that has been built over the site of the Chernobyl disaster that took place back in 1986, which is the year my parents were born. And this was in an effort to contain the continual radiation that was being released. They think that it will still occur for another 20,000 years based on the half-lives of the elements that were involved in that power plant. Then we also have biological agents, and these can be bacteria or viruses, which insert their genetic material into our genome. And although there's a couple of examples here, this is only an extremely small sample of the types of mutagens that can cause mutations in our genome. So, with that being said, let's look into some questions together. Here's a multiple choice question taken from, again, QCAA, and I've put a definition here and an image from the textbook to help you answer this question. The question is, which of the following processes would result in aneuploidy? So pause the video here and we will go through it in a moment. Okay, I hope that you've had some time to think about this. So, which would result in the presence of an abnormal number of chromosomes in a cell? Would it be A, the exchange of genetic material that occurs during crossing over? Well, crossing over does result in variation, but aneuploidy specifically results from this non-disjunction. So crossing over isn't going to cause that. It may cause inversion or other kind of chromosomal changes, but not non-disjunction. B, the failure of a homologous pair to separate during meiosis 1. Okay, well that kind of answers that. That looks like a good option. C, the failure of chromosomes to correctly align at prophase 2. It's not referring to non-disjunction again, and either is D. So the answer in this case is B. Next question. So, during oogenesis in a human, sister chromatids of the X chromosome, so we're not talking about homologous chromosomes, we're talking about chromatids here, fail to separate, resulting in the ovum containing 2 X chromosomes. After the ovum is fertilized by sperm with a normal chromosome number from the male, which of the following would represent the somatic number of, and these are the somatic numbers by the way, of the zygote formed? Pause the video here. Okay, so is it A, 2n plus 1, B, 2n? No, we know that's normal. Is it going to be normal take 1, or normal take 2, or is it normal plus 1? So it is, once by the way, if we have haploid come together, that's what 2n is. But in this case, we have an extra X chromosome, so it's going to be 2n plus 1. The answer is A. Let's look at another question. So we have Let's go there. Short response question. Looking at mutated strands of bacteria, a wild type, WT, of bacteria and 2 mutant strands, M1 and M2, that had disrupted genes. So they looked at the maximal growth rate to determine the success of these mutant strands of bacteria. And the results of the experiment are shown. We're looking at competitive ability, growth rate, and maximal growth rate. Here's the questions. Draw conclusions about the competitive ability and growth rates of the wild type of bacteria used in this experiment. The second question is, appraise the outcome of the data. Appraise, remember what that means. Think back to going to Gatton. Of the data and determine the success rate of the mutations for this species. So pause the video here and give that one a go. Okay, let's go through the exemplar answer scheme together now. So here it is. I'm learning that this is the more important section of the exemplar. This is just a sample response showing what you might have got marks for. But here's where the marks are. So for A, draw conclusions. You get 1 mark for identifying that the error bars overlap here, and therefore there is no difference in results. So if you are familiar with error bars, it's saying, well, there's a bit of an uncertainty. The answer could kind of be anywhere in between here. There's variation in the results. And a bit of uncertainty, it could be up here. So if we have a look, like if the answer could be down here or up here, it's all pretty similar for M1, M2, and the wild type of bacteria when it comes to competitive ability. So you would get a mark for that. And then providing a conclusion from the data about competitive ability and growth rate being similar based on that. And there's an example explanation. So B, it's asking you, let me just check the question, appraise the outcome of the data and determine the success rate. So you get 1 mark for identifying that there was no significant difference in maximal growth rate. And we can easily see that, but you can refer to, again, the overlapping error bars there if you like. You get another mark for identifying that the mutations do not provide a selective advantage. So appraise the outcome of the data and determine the success rate. Does this do that? There was no significant difference in the maximal growth rate between mutant and wild bacteria. The mutations did not provide a selective advantage. Okay, let's do one more question together. And it is 2 marks. Explain. So how many sentences did we say for explain? How damage caused by UV radiation can cause mutations in DNA. Let me give you a bit of context to this. So when you see that cognitive verb, this is what it actually means: to investigate, inspect or scrutinize, to consider an argument, to uncover assumptions and interrelationships of the issue. Basically, when you see the word explain, think 7 or 5 to 8 sentences would be fine. So I want you to research this and come up with a response. Pause the video here and I'll show you an example in a sec. Okay, so 2 marks. You get a mark for explaining how changes in DNA structure cause mutations, and another mark for explains how breaks in DNA cause mutations. So here's the sample response. UV radiation changes the DNA structure, it bends the DNA, causing the DNA to be read incorrectly. And UV radiation does this. Cancer is an example. Okay. Last question. This one more. This is it. Describe how inherited mutations can alter the variations in the genotype of offspring. So there's our keyword. What does describe mean according to QCAA? It means to give an account of a situation, event, pattern, or process, characteristic features of something. So define 1 to 2 sentences, describe 3 to 4 sentences, explain 5 to 8 sentences. I want you guys to research this and come up with the response. Pause the video here and show me what you get in class. Here is the exemplar response. You get a mark for saying that genotype variations identifies that inherited genotype variations are permanent alterations in the DNA sequence. 1 mark for identifying that inherited genotype variations can be none, small, or large. And a mark for identifying that inherited genotype variations change control genes. Okay, here's the sample. An inherited genotype variation is a permanent alteration in the DNA sequence. Variations can cause no change, a small change, or a large change in phenotypes, and changes in control genes can cause major changes in body morphology. So control genes, we're thinking back to gene expression and gene regulation. That's the HOX gene, right? From last week, that results in or determines the morphology of the body. Okay, well, that is all for today. And I will leave you with this quote by this man, Francis Collins. We've looked at him previously, right? He's been credited with cracking the genome. And he says, all illnesses have some hereditary contribution. It's been said that genetics loads the gun and environment pulls the trigger. So I just wanted to finish with that quote and saying, in light of everything we've spoken about, mutations and mutagens, that we do not have to be afraid or worry about the things that we cannot control. But rather, I see this as, let's make the most of the things, the opportunities, our environment, our choices that we can have influence over. I hope that this has been informative and helpful. Please watch over this, do your textbook readings, guys, and I will see you in class. God bless.